Hi, this is Miss Litton, and this is an Honors Bio Throwback Unit 2 review for the final. Say hi. Hi. Right. So we need to go over some things here, maybe take notes up. Another thing I would do is I would, maybe in a different color or something, you write like, I didn't remember that very well. I need to go over that some more, okay? And make note of that. Stuff you've got down pat, you don't need to worry so much about. So let's just go through these and refresh our brain on all of these different topics best we can, okay? So the first one, characteristics of life. Here we go, ready? Living things are organized, made of cells. They acquire materials and energy. They maintain homeostasis. They respond to stimuli and reproduce. They grow and develop, and they adapt and die. Okay, and organization. Remember organization? Atoms, molecules, organelles, cells. Why do we do this? Life starts, okay. Cells, tissues, organs, organ systems, organisms, population, community, ecosystem, biosphere. Yay! Classification of life. First of all, the classification categories. Let's go over those. Ready? Remember, what's the mnemonic for that one? Did Philip come over for good soup? Ready? Here we go. Domain, kingdom, phylum, class, order, family, genus, species. All right. What were the domains? What domain are you a part of? Eukarya. Okay? Then you are part of eukarya because you have what kind of cells? Eukaryotic cells. What are the other kinds of cells? Prokaryotic cells. There are two domains too within the prokaryotic realm. What are the two? Bacteria and archaea. Exactly. I'm tired. Bacteria and archaea. They're both prokaryotic, but they have different membranes, different walls. Who's more closely related to the eukaryotes, bacteria or archaea? Archaea. Remember, they evolved later. 99.9% you get the right answer. So don't worry about that one. So archaea are more closely related in their ribosome structure. They branched off later. They were misnamed. And they were named archaea because they thought they were really ancient. But it turns out they're newer. Okay? Bacteria are found everywhere. Archaea are found, they're called what? Extremophiles, found in real extreme areas. And then we even went over man's structure, or man's classification. Remember that one? We are what domain? Eukarya, right? Domain, eukarya, right? Domain, kingdom, Animalia. Phylum, Chordata. Domain, kingdom, phylum, class. Look who's in the class. A bunch of mammals, mammalia. Domain, kingdom, phylum, class, order. Primates, because we remember what? No monkey. Order, family, hominidae. They're hominidae. And then genus, species, right? Would be what? Homo sapiens. Perfect. No, not Java. Okay. We did it. Okay, atomic structure. Here we go. The atom story. Atoms are made of protons. Wait a second. Atoms, just a minute. Okay. A second, I have to write myself a note. Okay. Maybe. Okay. Atoms are made of protons, neutrons, and electrons. Did you know that? The protons and the neutrons are in the nucleus, atomic mass. The electrons are in energy levels. 2, 8, 1832. 8 is great octet rule. Perfect. Ions versus isotopes. An ion is when you vary the number of electrons. Isotopes is when you vary the number of neutrons. What's bigger, an electron or a neutron? it gets the bigger name, the isotopus. Okay, it gets the bigger one. Ion is very little, it's an electron. What happens if I gain electrons? What will happen to my charge? Negative, but I gain energy too, right? By the way, who would give me electrons? Hydrogen. And when I get his electrons, what do I become? Reduced, and he becomes? Just saying. Okay, bonds. Ionic is when you do what? Are you sharing? No, it's all about yourself. Ionic. It's all about me. Okay, so you will ditch or steal. Gain. That's right. That's chemistry. I know, I gotta personify it. Ditch or steal electrons, right? So your charge is going to be not neutral anymore as it shows up on the periodic table. And remember we talked about what are the odds of ionizing. Remember that part? If you have, what would make you very stable and that you don't want to ditch or steal any electrons? In two ways. Last column, which means your what? What makes you that? Your outermost energy level is full, or it's at 8, hence the octet rule. Okay? If you have your outer energy level full, or it's at 8, then you are stable. Okay? Full, okay. And then if not, you will tend to either gain or lose electrons in order to have that stability. If you ditch them entirely or steal them, then you will have a charge and you will enter into an ionic bond. Right? Because opposites what? Attract. If you share to get stable, that would be a covalent bond. If you share the electrons evenly, perfectly, then that would be called a non-polar covalent. If you share electrons with someone unfairly, so one has more than the other, that is called polar covalent. Okay, ionic covalent. Oh, hydrogen bonds. We know about hydrogen bonds. That weak force, right, between one partially charged molecule and another partially charged molecule and atom in there. So a good example of that we talked about hydrogen bonds was with what? Water. And the oxygen is slightly what charge? Negative, and the hydrogens are slightly positive because oxygen is very electronegative and pulls on hydrogen's electrons. So now this is, my hands would be positive and my back would be negative. So my hands, right, my positive hands would be attracted to another molecule's what? Another water molecule, her what? Back, right? Am I touching her? Am I sharing any electrons with her? No. It is a weak force that you see in water molecules, and you can do that with any polar surface. Okay, that weak attractive force. Where do you see hydrogen bonds really important to us? Besides water. DNA, holding the DNA together. Yeah, crawling up plant xylem, sure. And we talked about, does that come later? No, let's talk about it right now. Properties of water, right? Remember that? Yeah. Properties of water, it's a liquid instead of a gas, right? It's a good solvent. It has, remember we talked about cohesion and adhesion? What is cohesion? What? Water molecules with itself. Adhesion is when water is attracted to another polar surface. High heat capacity can hold a lot of energy in those hydrogen bonds. High heat of vaporization takes a lot of energy for it to go from a liquid into a gas. Ice, good. Less dense than liquid water, so it floats and becomes a protectant for the life below. It insulates. And then we talked about one more, well actually it's, you can cool yourself off with that high heat of vaporization, but there was another one I wanted to talk about. Adhesion, gosh, why aren't I thinking? It was just in my mind. Adhesion, cohesion. We talked about transpiration in plants. Mediating effect of ocean water, right? Because where you have water, it can absorb the heat all during the day, and then at night it can re-radiate that heat back out. Okay, acids and bases measured by pH. Good. If you're an acid, you have a lot of what? Hydrogen ions. A lot of hydrogen ions. And your pH is low or high? Low. Acidic. And the opposite of that, you have fewer hydrogen ions, more hydroxide ions, and your pH is high. Bleach is a base, right? Hydrochloric acid is an acid. Lemons are a acid. Tomatoes are a soda is a acid. Okay. Organic molecules, monomers and polymers, and how you build them. First of all, it's like we're doing like 2 months worth of work in a nanosecond. Okay, yay! Okay, so when you want to build, it's the same process. There's 2 names for it. What was the name? Dehydration synthesis and condensation synthesis. Dehydrate. If I dehydrate something, what am I doing? I'm pulling away. I'm pulling water away. And I'm doing that because I'm removing an O8. I'm pulling an OH from one, right? And just H from the other. And then these will go and be what? Water. And then these will bind together. Never done that in class? There's probably a reason why. It's okay. You know what I mean. Okay? And then hydrolysis is... Hydrolysis is when you use water and you break them apart, right? Yeah, hydrolysis. And you give an OH to one and an H to another, and then you break it apart. That's the same way all the time for building. 4 important molecules. Think them in your head. I'm thinking them in my head. Now think what each is built out of. Think it in your head. See if you can come up with it all by your onesie. All right. First one I talked to you about was what? Carbohydrates. What are carbohydrates built out of? Sugars. Sugars are a form of energy. You can take simple sugars and form disaccharides. Okay? Disaccharides like maltose, which is made out of glucose and sucrose. Your table sugar is called sucrose. It's made out of glucose and fructose. Fructose is fruit sugar. Okay, milk is lactose, made out of glucose plus galactose. They all have glucose in them, don't they? GG makes maltose, right? Glucose and galactose makes lactose, and glucose and fructose makes sucrose. Good. Those are disaccharides. They're for energy. Then you can have polymers, where you hook a bunch of sugars together. Also, you could form energy. Things like starch is energy, right? And you would find that in a plant. Or what? How do we store that energy in our liver? So do you remember what it's called? Glycogen. Okay? But polysaccharides can also be structural as well. They can build cell walls. They can build exoskeletons in bugs. And that would be like cellulose, chitin, are structural. All right, carbohydrates, check, done. Lipids. Lipids, 2 categories. Fats and steroids. Steroids are 4 rings of carbon, and you can have things like hormones, right, that are steroids. Cholesterol is a steroid. Not all hormones, but there are steroid hormones like testosterone and estrogen and progesterone. Fats are made out of 2 components. Do you remember what they are? Glycerol, which is 3 carbon unit, and then fatty acid chains, long chains of carbons and hydrogens. And we know we can snip down those long chains of carbons and hydrogens and put them in cellular respiration and get a bunch of energy from them. Hello, sweetheart. And that's why fats have more energy than carbs, right? Okay. Just passing through. Oh, you just need to work a little bit.
I just need the computer real quick.
No problem, sweetheart. Okay, so, let's talk about those fatty acid chains. They can either be or unsaturated. Where do you get the saturated ones from? Animals. And they're what at room temperature? Solid. Plants are liquid at room temperature. Good, okay. And then there's a thing probably on trans fats where they take plant fats and make it into a solid so you can spread it, but then it makes it really bad, so you might as well have butter. Okay, we did that, we did that. Okay, proteins. What are proteins built out of? How many different amino acids are there? Because you have 20 different R groups. Good job. Okay, you have a carboxyl, an amino group, a hydrogen, and then that R group. I don't know if you remember all of it. Do you remember that? I know. An amino group is NH2 or NH3, a carboxyl C double bond O, C, OH. Hydrogen, carbon. All right, fourth important organic molecule are nucleic acids. Give me some examples. I gave you 3. Yeah, deoxyribonucleic acid, DNA. Ribonucleic acid, RNA. And ATP, energy. And nucleic acids are always built out of what? Nucleotides. And nucleotides are always built out of what? Base, sugar, phosphate. Okay, and then ATP is adenosine triphosphate, adenine ribose, phosphate, phosphate, phosphate. Okay, now look to me. I rattle those off because I've been teaching it for 30 years, right? That's what I do. If you are not rattling them off in your head like I'm rattling them off, that's okay, right? You just need to review them a little bit and have a good idea. On the very basis, I would know the names of the 4 important organic molecules and what each is built out of, and I want to be able to recognize it, right? I'd want to be able to recognize that ring, the sugar, how it, remember we looked at it and we built it, break it in half, right? I want to be able to recognize a steroid, which is 4 rings all hooked together. I want to be able to recognize that fats are glycerol and long fatty acid chains and how they have a lot of energy because you know about cellular respiration. I'd want to know the structure of DNA because you do need to know the structure of DNA, and it's made out of nucleotides, base, sugar, and phosphate. How many different bases are there out there? 5. 4 of them are in DNA, adenine, thymine, guanine, and cytosine, right? Remember that part? What do we find in RNA that you don't find in DNA? Uracil. Okay, breathe deep. Remember how honkin' and massive that chapter was? It seemed like we were doing it for like never-ending hell of days of lecture, right? And so it was like day after day after day. So that was a big chapter, big, big chapter. All right. We already did 5. Now we're on 6. We already talked about 6, basically. Prokaryotic, eukaryotic cells. Which one is smaller? Which one is like a one-room schoolhouse? Which one's like a college? Exactly. All right. Structure of cell membranes and the functions of phospholipid bilayer, right? And then you have interspersed among them proteins, right? Some can be peripheral, some can be integral. On the outside, you could have the glycocalyx, those carbohydrate side chains. They're either called glycolipids or glycoproteins. Good. On the inside, you have the cytoskeleton. Okay. Let's talk about the function of the proteins in the membrane, shall we? Okay. They can be channels, carriers, cell recognition, receptors, enzymatic, and cell junctions. It's called a phospholipid bilayer, and it's semi-permeable. Like some things can pass through and some things cannot. So remember we talked about the 3 ways to cross the membrane? You can go through the phospholipid bilayer, but not if you are large or charged. Or you can use a channel or a carrier or whole membrane. Endocytosis, exocytosis. Let's talk about through the phospholipid bilayer. I'm talking CO2, O2, water. Oh, let's focus on water. What is it called when water passes through a cell membrane? Osmosis. And osmosis is the diffusion of water across a semi-permeable membrane, yes? Okay. Then I think of words like hypertonic, hypotonic, and isotonic. You guys remembering all of this? Yeah. Of course you do. Hypertonic, hypotonic, and isotonic. Water must flow from the hypo. Exactly. Right? Okay. So how did a cucumber become a pickle? It was put in a what? Hypertonic. And if it's a sweet pickle, it was put in water that was very, very sugary. So the water around that pickle is hypertonic. Oh, sorry, around the cucumber is hypertonic. The cucumber is hypotonic. Water must flow from the hypo, flows out of the cucumber and where? Into the sugary water, and the cucumber gets smaller and smaller and smaller until it's a pickle. Right? Okay. That's through the phospholipid bilayer. Then you could use what? Channels, carriers. If it costs you no energy and you use a protein to get through, what is it called? Alters in that. Facilitated diffusion. You're just going from a higher concentration to a lower concentration using a channel or a carrier. If it costs you energy, it's called active transport. What was our best example of active transport?
The sodium potassium.
Sodium potassium pump, baby. All over it. Good. If we need to use the whole membrane, it's called what? Endocytosis and exocytosis. In endocytosis, there are 3 types of endocytosis. Cell drinking, which is called what? Penocytosis. Cell eating, which is called phagocytosis. And then, ding, 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 ding. What's that? Receptor-mediated endocytosis. Do all of those cost energy? Yes. So what's it called? If you're moving a membrane all over the place, what's that called? Active transport. We totally did it. Yeah, look at the rest of them. Do you have any questions over those that you would like me to? Do you want to have a little secret for your final? Yes. Your secret. I know a couple of additional objectives really well. I'm going to let you pick which one you want to talk about. Okay. What are additional objectives? I love you so much. Do you have any other questions? No, the ones that I italicized that I asked you about other. Oh. Oh. I didn't just say.
So additional objectives. That's what we're supposed to know in the final.
Yes, that's why I would think you have to know. I would just be a specialist. Okay, is there anything else I can help you with? Okay, you're simply. I know. Have good weekends, make good choices. When you can vote, use your voices. All right. Have a piece of toast, all right? I hope you're studying. I hope this isn't the night before your final. I'm very disappointed if this is the night before your final and you're listening to me now, though I'm glad you're listening to me now. But we want to tell them next semester they need to do what? Make better choices. But we love you either way. Okay, later.